have one objective this evening, and that's to convince you that the smallest things in the ocean really do matter, and in particular, the microalgae that photosynthesize. In other words, they're plants. And throughout the next 40 minutes, I'm going to try to show you, give you some insights into why these tiny little microorganisms make such a big difference to the planet that we live in. So this is quite a deliberate photograph to be provocative at the beginning. Our planet is down there in the bottom right-hand corner, and center stage is Cosquina discus. It's a large diatom, pretty ubiquitous around world's oceans. The yellow bits are actually the chloroplasts. That's where the photosynthesis takes place. And the beautiful sort of framework that you see is the skeleton made of ultra-pure silica. And I'll talk more about this as we go forward. So just to orientate you through this evening, it looks like I've got quite a lot of things to cover, but they're in about 4 main groups. I want to give you a sort of introduction at the beginning and try to allow you to see these microscopic organisms, tell you a little bit about how that is achieved and some of the history that has gone into our understanding. Then we'll get into a little bit about why they matter, particularly their role in the health of the planet, what we call the biogeochemical cycles. And then, as Andy hinted, I couldn't pass up the opportunity to tell you a little bit about what we do at the Bigelow laboratory and some of the advances that we're doing. I've got a few slides just in the middle for the die-hard scientists that might be in the audience who want to see some science before it even gets to a publication. And I should have mentioned right at the outset, I've already been remiss in this, my co-author is Mike Lomas in this. I'll talk more about Mike as I progress through. He also is the director of the National Horticulture Facility at Bigelow, but I'll explain that in the talk. And some of the science here is Mike's work that he's working on right now. And then really some provocative questions and some issues around responsibility for using this wonderful marine resource, both here in Maine and in the global oceans. And I've got a little bit of publicity right at the end to whet your appetite about an exhibit that's not far from here up in the Portland Library. Okay, so this is how most people think of the oceans, particularly this time of year, with a bit of sunshine, nice clear water, nice blue, calm seas, and so on. And we don't think much beyond that. We might, if we're in a location like this, get our swim fins on and our face mask and snorkel and have a snorkel around. But most people never go very much below the top 10 feet of the ocean. And yet it covers 70% of the surface of the planet, and 98% of all water on the planet is locked up in the ocean. But within them, it's teeming with life. We know about the fish and the whales and the dolphins and the turtles and the coral reefs, and we know about the lobster, and we probably know a little bit about oysters and mussels and clams and things that we like to eat. But up until just over a decade ago, maybe 15 years ago, we did not know the most common organism on the planet. This is it. It's not particularly stunning. In fact, it's pretty small. That scale bar is 10 microns in size, which is 1,000th the diameter of a human hair, so it's pretty small, these things, but they're extremely common. It's what's called a cyanobacterium. It is actually a bacteria, but it photosynthesizes. Its name is Prochlorococcus marinus, and it was discovered through some of the techniques that I'll talk about today, namely flow cytometry. And Penny Chisholm at Massachusetts Institute of Technology, not far from here, is credited with doing the early work on this, working with people at Bigelow and in other places. So this is really one of the tiny giants in the sense that it is performing a very important process. It is taking carbon dioxide from the atmosphere and fixing it through the process of photosynthesis, which I'll explain. But let's follow this theme of size. So the mountaineers in the audience will recognize Everest at the backdrop to this, and that's somebody walking over the top of Everest. And the point about this is if you were to make one of those microscopic plankton the same size as a human being, it would be able to step over the top of us. Or, another way of thinking about size, if you shrunk a human being to the size of one of these microplankton, you would fit the entire population of the planet into a shoebox. So that's kind of little ways of thinking about just how small these are. But of course, we also need to think about how many of them there are. We'll come to that in a minute. Here's this photosynthesis. This is the only chemical equation that will be up here tonight, but it is an important one. It is actually pretty much what drives all life on the planet. We have had some exciting discoveries over the last 20, 30 years where there's life coming from chemical sources and chemical energy deep within the Earth. But by and large, the whole food chain on the planet depends on this process of taking carbon dioxide in the atmosphere and water and using photons, light, to drive a chemical reaction, which produces, well, in this case, it's organic molecules, as simplified here as sugar, and producing oxygen. And it's the oxygen, of course, that the animals on the planet need to respire. And there is so much marine phytoplankton in the ocean performing this photosynthesis reaction and producing oxygen that every time we take a second breath, we're breathing the oxygen that has come from marine phytoplankton. The other half is coming from the forests and on the land. And these microbes are performing a really important and valuable part of life on the planet. It hasn't come out terribly well. I thought we needed to have a picture of the Gulf of Maine, and we needed a winter picture. So you can see Nova Scotia, and you can see us just about here, I guess. And because it's a winter picture, the phytoplankton are not particularly blooming or growing, but you can still see some over in the Georges Bank here. You see Cape Cod, so you can see some around the coastal areas. For any geologists in the room, that little impact crater at Lake Manicougan stands out really well. That was a meteorite that struck Canada that left that there. So we can obviously start to see these tiny little microorganisms from space. There are so many of them that satellites enable us to see them. One of the founder for Bigelow Laboratory was the late Dr. Charlie Yench. And Charlie was working in the 60s and 70s with the very first satellites that NASA was putting up that was able to image the color of the ocean. And he worked on transferring that color imagery into a quantitative measure of biomass, in the sense that the color is the amount of pigment or chlorophyll that is in those phytoplankton. And because the satellites were orbiting the Earth, they could essentially do that over and over again and produce a composite image of the distribution of this microscopic plants in the very surface layers of the world's oceans. So what you're looking at is actually the vegetation on the planet. We know it well in the rainforests of South America, the boreal forests of North America and Europe. But if you look in the oceans, you can see color, well, this is a false color image, but you can see changes between the so-called central gyre areas, or the deserts of the ocean. This would be analogous to the deserts, to the areas that are very much more productive here in the equatorial Pacific due to the upwelling, and particularly the temperate regions where there are more nutrients that come into the surface. So in a sense, 30 to 40 years ago, we began to see that the distribution of these microscopic plants in the ocean was could be described, and we could start to understand that in terms of the supply of nutrients and circulation in the world's oceans. And we also began to realize just how much of them there were, because we could perform calculations, understanding the rate of photosynthesis, to start thinking about how much carbon dioxide was actually being taken up into the world's oceans and turned into biomass. And this biomass we call primary production, and that is essentially the base of the food chain for all marine and aquatic life in the oceans, and of course in lake systems as well. But these microscopic plants come in many shapes and forms. Indeed, there is more diversity in the microbial world, in the world's oceans, than any of the diversity that we're familiar with on land. If we use things like genetic measurements to understand the diversity of life, then there is far more diversity, way more, in the oceans, in the microbial world than exists in the larger organisms that we're familiar with on land. And I don't have time to go into all of these, but I will show some important classes of those in a moment. But I just wanted to illustrate some of the variety of shapes and forms that you find, and sizes, of these microorganisms. Okay, so a little bit back to some numbers, because I like numbers in the sense of trying to understand how these things matter. So let's take some small phytoplankton ranging between 1 and 100 microns, and if you took 1,000 of them, put them end to end, you should just about make the thickness of 1 cent of a penny, and about 18,000 of them would fit across the face of the penny. But if you took a can of Coke and filled it with seawater, you'd have something like 75 to 100 million cells in one can of Coke. So there's incredible numbers in small volumes. In terms of the biomass of the Earth, these small phytoplankton only make up about 1% of the total biomass on the Earth, but they're accounting for nearly half of the photosynthesis, and particularly this reaction of taking up carbon dioxide and creating biomass. So they're extremely important. So I've tried to convey to you the sense that these microorganisms, these phytoplankton, are extremely abundant, and they're essentially creating a living planet through the production of oxygen and themselves changing through the seasons. So now we want to try to kind of investigate them a bit further and try to understand how we're able to see such small things. It's hard actually to find the very first person to make detailed descriptions of marine phytoplankton, but probably one of the most famous is this fellow, Ernst Haeckel. A very interesting fellow. In a moment I'll point you at a film that you can see that sort of illustrates his life story. He was one of these people who could never really decide whether to be a scientist or an artist. And he moved between those two extremes considerably in the course of his career. And just to give you an example of his artistic capability, he was looking down the microscope and drawing diatoms that he could see down the microscope. And this compendium covers all types of marine life, not just the diatoms. You can see jellyfish here, and he was describing corals and seaweeds and all sorts of things in his investigations. I mentioned a film about his work, so you can look this up on Google. I might even be able to download it, called Proteus, which essentially takes some of the images that he drew and weaves it into the story of his life, and it also animates it and sets it to music. So it's a good way to de-stress and learn a little bit of marine microbiology at the same time. So Haeckel was using this. It's a pretty primitive microscope, a light microscope with a few objectives, just about powerful enough to see some of the larger marine diatoms that he was drawing down the microscope. But of course, as you've already seen some of the images that I've shown you, we have some pretty advanced technology these days. We still do use light microscopes, albeit much more advanced than he had, and usually with camera systems attached to the top. So that's still dependent on photons being transmitted through a slide where the microalgae are held in suspension. We can go one step forward and fire laser beams at the sample. And with lasers, we're also exciting the sample, particularly if we've put dye into the tissue of the small organism, or indeed the tissue itself, if it contains organic material, can autofluoresce. So we can get a different type of imaging using what's called a confocal microscope using laser beams. And if we want to go another step forward, we can fire electrons at the sample and essentially create an image from the way in which electrons are scattered off the surface of the sample. And some of the images that I'll show you tonight have come from this thing, a scanning electron microscope, and some of them have been created with light microscopes and confocal microscopes. So we are able now to essentially take very detailed images of things that are 1/10,000th the diameter of a human hair. So that is an amazing ability to sort of image these microscopic organisms and see the diversity that exists there in the ocean. So here's a couple of examples. This one is back to a diatom. This is using that scanning electron microscope. And what you're looking at is the skeleton of the organism. The soft tissue has disappeared, and you're just left with the skeleton, in this case, made of pure silica. The actual mineral phase is opal, just like the semi-precious stone. It is hydrated silicon dioxide. And in this case, the microorganism is making these discs, which it builds into a skeleton, forming a chain. So it can form a long chain of the microscopic plant. So that's the case where the skeleton is made of silica. And here's a different one, but this time the skeleton is made of chalk or calcium carbonate. This is a promesiophyte marine microalgae. Its skeleton here is made of liths of chalk. So we often refer to this as a coccolith. That's a sort of a generic name for it. And the Latin name for this one is Emilianae huxleyi, which is, again, a very ubiquitous marine algae with a skeleton made of chalk. This one, because chalk is quite reflective in ocean water, is one of the ones that we see very easy from satellite images. And particularly towards the end of a bloom, when the soft tissue is starting to decompose, and these plates of chalk get sort of broken up and scattered in the surface waters, the water becomes quite reflective to light. And sometimes when you're on a ship sailing through a patch of water like this, it's actually turquoise in color, the same color as a lake in the mountains that's fed by glaciers, that same sort of turquoise color. So we have one class of these marine microscopic plants with skeletons made of chalk, and another one made of silica. There are yet other ones that don't have such major skeletal compound production, but this is extremely important in terms of the chemical cycles of the planet. You can accumulate so many of these skeletons of these microorganisms on the seafloor that over geologic time it will build up to form rock layers. So that often the chalk that you'll see, if you reflect back to Britain, where I'm from, the white cliffs of Dover are made of the skeletons of these coccolithophores. If you go to California and you look at some of the sediments you'll see there, they're probably made of the diatoms, the silica-rich ones. So there are so many of these skeletons accumulating that they form geologic deposits. Now, just a little bit of how things are changing on the planet and what the scientists are thinking about is happening. Thought I'd put up one or two sort of controversial things for you. The first one is this paper that's published a little while ago now, 2010, by Boyce, Lewis, and Worm, up from Dalhousie in Halifax, Nova Scotia. And they essentially looked back over rather simple records. The records were the way in which the transparency of the water has decreased over time using the same measurement, something called a Secchi disk, and that measurement has been repeated many times world over, back over nearly 100 years. And when they've done that analysis, they argue that in certain ocean basins you can see declines in the amount of the phytoplankton over the last century. A figure of as much as 40% since 1950. This is debated, and people like Andy work on these kinds of data sets and others to try and build our predictive modeling capability going further forward. But we do know that one of the things that is happening is that the carbon dioxide that these microorganisms are using to photosynthesize also dissolves into the ocean water. And just like any time you put carbon dioxide into water, it tends to make it acidic. So this is just a little cartoon showing the carbon dioxide in the atmosphere dissolving into the water and undergoing what's called dissociation, essentially into different acidic species that exist in the water. And of course, anything that's then made of chalk tends to dissolve in water that becomes more acid. So I mentioned that some of these small microorganisms, these coccolithophores, have their skeletons made of chalk. So they're obviously going to struggle and suffer under ocean water that's becoming more acid. Many other marine organisms have skeletons made of chalk. Coral reef systems, bivalve mollusks like oysters and mussel shells. All of these things will essentially have some kind of response to water that is becoming more acid. Just to show you how that is happening over time, this red curve increasing upwards from about 1955 up towards the present day is the amount of carbon dioxide in parts per million that has been measured on the summit of Mauna Loa, the big volcano in Hawaii. So we've been essentially making measurements of the atmospheric carbon dioxide over the last half century. This is a fairly old slide, I'm sorry. It goes up to 2014 now, obviously, and we have now exceeded this 400 parts per million in atmospheric carbon dioxide. As the CO2 increases in the atmosphere, it also increases what's called the partial pressure of CO2 dissolved in the water here. And so this is the within water measurement, and you can see that it parallels the amount of CO2 in the atmosphere. And the consequence of that is to make the pH, or the acidity of the water, diminish. You can see a pH scale here on the right, and you can see that that's declining. And this is in the open ocean of Hawaii. This process is exacerbated in coastal waters, and it's very important here in the coastal waters of Maine, which are cold. When waters are cold, we actually get more CO2 dissolving into the water. And so Maine is one of the first, well, it's the first state on the east coast of the US to essentially set up a commission to understand the impacts of this process called ocean acidification. And there's a bill currently in front of the legislature to enact an ocean acidification bill for the state of Maine to try to bring together the knowledge about what this process is and what that means for our fisheries and our ecosystems in the coastal waters of Maine. So, just a little summary here at this part of the talk. Hopefully I've convinced you that these marine microalgae are doing a very important thing called photosynthesis. Put some numbers on that: 105,000 million tons of carbon per year. That's removed from the atmosphere and turned into biomass in ocean water. And already, you guys in this room have probably breathed around 500 times since you've been sitting here listening to me, and 250 of those breaths came from marine phytoplankton. So we can thank them for us all staying alert. And then we can see that they're at the base of the food chain. That's important for the health of things like the fish ecology. And there are implications for human health, which I'll come to at the end. Okay, so now I'm going to go on to talk a little bit more about what Bigelow is doing with these marine phytoplankton. These are the laboratories that Andy mentioned in his introduction for me. And we like to think of the science that goes on at Bigelow as being sort of global in scope and interdisciplinary in nature. So we're working on issues of the entire climate system of the planet. We think about the relationship between climate and the health of the oceans, particularly these microbes that live in the world's oceans. And that has an intersection with something called blue biotechnology. It's just a fancy name for applying the knowledge of biotechnology to the ocean or marine environment. And this is another way of thinking about looking at the genomics and biochemistry of the cells of the marine microbes in the world's oceans. So we at Bigelow espouse discovery. We're just like the GMRI, it's an independent research institute. We're interested in acquiring and discovering new things about the world's oceans. And I think that fits very squarely with oceanography as being a science of discovery. We still don't know very much about the oceans. So whilst we're trying to apply what we know, we're also spending a great deal of time just trying to understand and find new things. We work on all this, all maritime nations have always conducted voyages of discovery, sometimes for long periods of time, like the Census of Marine Life or the International Census of Marine Microbes, which is that acronym there. And I've just got a little cartoon. This was back to the Challenger expeditions of 1885, 1884, 85. You took your samples and put them in a bucket, and you had a dog, and you looked into the bucket, and you discovered new things about marine life. Here's some of Ernst Haeckel's drawings, and now that is just a way in which we represent the genome of a marine microorganism or its genetic information. So we've gone from looking into a bucket to actually identifying the complete genome of a marine microbe in just about 130 to 140 years, which is pretty amazing, actually. Bigelow scientists go the world over, here up in the polar regions, coring through the sea ice to obtain marine samples, to some nice weather in the tropics, collecting water samples in these bottles here from different depths in the world's oceans. Sometimes we participate on circumnavigation cruises, so that sort of spirit of adventure is still alive and well. This is the Tara expedition, and I think one of your last speakers of the Sea State Lecture series, Lee Cart-Boss, is going to talk about more work from the Tara expedition. This just shows you some of where Bigelow scientists have been just recently, all different parts of the world's oceans. Okay, so I'll just going to illustrate our capability in these marine microbes by just describing some of these sort of facilities that we have. Starting with the culture collection, talk a little bit about the role of flow cytometry, and then particularly the single-cell genomics center. I'm going to just kind of roll these together as capabilities for understanding more about these marine microbes. So starting off with the NCMA, the National Center for Marine Algae and Microbiota. This is designated by an act of Congress back in 1995. It's the US National Repository. Mike Lomas is the director of that facility. And it is probably the world's largest, most diverse collection of these marine microalgae and other microorganisms. So there's a lot of Latin names here, but it just shows you the sort of variety and distribution of the types of these organisms that is held in the NCMA. And the unique thing about a collection like this is it's living. This is not like dried flowers pressed into books and kept on a shelf. These plants are living. So they all have to be in individual test tubes of culture medium. They have to be grown. They have to be looked after, nurtured, propagated. You have to speak to them nicely every morning, make sure they're happy, got enough light, got enough warmth, all those sorts of things. Some of them we are a bit more brutal with. We just stick them in liquid nitrogen and freeze them to, not death, we freeze them to suspended animation. And then we can bring them out of the deep freeze and they're still able to be propagated. So we have a lot of knowledge and skill locked up in the 40 years of existence of the National Culture Collection. And these are just images off our website. If any of you would like to see more about marine microalgae, do have a browse around the website, because it enables you to kind of explore, for instance, algae that might live in cold water, or ones that might have high lipids content. That's oils and fatty acids, or you might be interested in the ones that are toxic. So these are different types of things that you could explore through our website, and they would tell you about whether we have those organisms in the collection and a little bit more about them. They come from all around the world. Those of you with very good eyesight will see that some of these locators seem not to be in the ocean. And that's because the collection also accesses specimens from other institutions and research centers and so on. And sometimes we've actually not really 100% sure where that organism had actually come from. So the attributed location, it becomes a research lab rather than the ocean. But there is still many within the collection, from the tropical regions of the world's oceans through to the polar regions of the ocean. So the ones that come from the tropics, they need to be grown at 25 degrees centigrade, up at 70 degrees Fahrenheit, whereas the ones from the polar regions need to be kept cold, nearer 2 to 4 centigrade, 37 to 40 degrees Fahrenheit. If you want to explore the biodiversity in a collection, this is also on the website. So you can go through the tree of life and click on these different classes of the microbes and understand what's in the collection. Humans are down here in this, well, they're a branch further off the eukaryotic tree of life beyond the eukaryotic bacteria. So we're not even represented on this diagram. So I'm just moving into another technology, which is flow cytometry. And this is the ability to use a microscope, in a sense, but also to separate out the different marine microbes that might exist in a sample of water. So this is a cell sorter at the top here, which allows us to essentially image and create a distribution of the different types of marine microorganisms in a sample of seawater. This is the FlowCam instrument from Fluid Imaging Technologies, based just up the road. There's some members in the audience from Fluid Imaging Technology. That's partly a spin-out company from Bigelow Laboratory, which is now building instruments that go the world over. And pretty much every marine research institution in the world now has a FlowCam, or at least they should have. So that allows us to see images of these marine phytoplankton as they come through a sample of water. Here's your seawater, pour it into the machine. Well, not pour it in, but you allow it to flow through the machine, and it will continually take photographs very rapidly and identify and size the phytoplankton automatically as it goes through the instrument. So, assuming you can grow these things and also that you can image these things, how do we get at the genetics of them? Because if you take a little bit of Booth Bay Harbor seawater, there's Booth Bay Harbor in the summer in the background, and you look at 1 milliliter of the seawater in the bottom of that test tube, we know that there's a lot of marine microbes in there, because I told you that earlier on. But if we were to translate that to the amount of genetic information, we would fill up one of your hard drives, a terabyte of genetic information in just 1 milliliter of seawater. But really what we'd like to do is to understand which microbes that has come from. Now, one of the challenges with working with plants, does anyone of you who have green fingers, is that sometimes they end up looking like this. This is on my windowsill at home. So, you know, I'm not very good at looking after plants, that is for sure. This is growing different kinds of algae in at different amounts. But one of the things that you'd want to do if you were going to do the microbiology of these microorganisms is you need to grow enough of them so that you could perform the biochemistry or the genetics. But the problem is that we only know how to grow a small proportion. The culture collection at Bigelow only represents a fraction of the marine microbes out there on the planet. Unlike on land, where we know how to grow the vast majority of the plants that live on land, we don't know how to grow 98% of the microscopic plants in the ocean. That's a big impediment to doing science. However, we have a plan. And the plan is to use this image, this technique of flow cytometry, because we can sort these microscopic organisms using the flow cytometer. Like this guy, who's the star of our show, Costina discus, we can actually sort Costina discus, one cell, that one cell, which was only about 50 microns in diameter, into a little plate in a rack. And then we start to do a lot of chemistry and some sequencing, and we can essentially extract the DNA and sequence the DNA from that one single cell. In other words, here's the cell. We have actually mapped the DNA from that one cell. We had to make many, many copies of the DNA to be able to do that, about 1 trillion copies of the DNA. But we were able to take one microscopic cell and transform it so that we could obtain enough DNA to sequence. And this is the art and skill of the Single Cell Genomics Center, housed at Bigelow Laboratory, and this is the sort of flow of how that process is conducted. This is now a world-class facility for doing single-cell genomics, and it's enabling us to do a whole range of new discoveries that you could read about on the website there. And it's led by Dr. Ramunas Stepanauskas. And he's also conveying this through the scientific literature in terms of the breakthrough that this enables, not just for marine microorganisms, but essentially any microorganisms, including all the bacteria that live inside the guts of humans and other mammals, all of which has really important opportunities in advancing medicine as well. Okay, just a few things to close off in. I'm coming in the last 5 minutes. Bringing us back to Maine and the algae, marine microalgae, I think most people are aware of something called red tides. And what happens is that the shellfish we want to eat are in turn eating something else. They're eating these marine microalgae. And those algae themselves have been accumulating toxins, which are then building up in the food chain and accumulating in the shellfish that we want to eat. So at Bigelow, we've developed some of the analytical techniques to measure the quantity of toxins inside the shellfish. And we do that now routinely for the Department of Marine Resources. And then they're able to employ the methods of control and monitoring to either open or close shellfish flats and so on to support or to protect human health in that process. Just now, just a little bit of science here before I close off. This is some of the work that Mike is working on. And I think I've explained to you that the environment is kind of heterogeneous in terms of the distribution of this phytoplankton in the world's oceans. And hopefully you've seen that we can understand something about the genetics of these individual microbes. We've had for many, many years this sort of concept that the marine microorganisms exist everywhere, but somehow the environment selects which ones are going to be the most competitive in a way that we haven't fully understood. In fact, Mike reckons we have not really made that full connection, if I go back, between understanding physiology and genetics and this sort of selection process, which in turn structures the ecosystem. And because the ecosystem is involved in the biogeochemistry, as I was explaining at the beginning, there's a feedback to the environmental control. This is essentially the living cycles of the planet and trying to understand whether the environment is controlling the microorganisms or whether the microorganisms through this system are actually influencing the planet. It's an age-old question scientifically. But we have some new tools available to us. So for instance, with that flow cytometry, we can actually sort these very small organisms. Here was Prochlorococcus, the one that was the most abundant organism that I started right at the beginning with. And this is the ability, or the saturation ability to uptake phosphorus. There's a little diagram down here in the bottom, but essentially all living tissue, these microscopic plants, need phosphorus and they need nitrogen as well as creating their biomass. And the ratio of the carbon, nitrogen to phosphorus in marine microbes, in these marine microorganisms, is sort of standardized, something we call the Redfield ratio, which we all learn about in oceanography. Except that that is an average. And when we get down to looking at in detail at these small groups of microorganisms, we start to see that the Redfield ratio breaks down. And what Mike has been doing is looking in parts of the world's oceans where the phosphorus content is low. This map, this color contoured map of phosphate here shows low levels of phosphate in the Atlantic or in the temperate North Atlantic, but higher levels of phosphate in surface waters in the North Equatorial Pacific. And when you look at the different microorganisms, you can see that their propensity to uptake phosphorus actually changes dramatically, both between the types of microorganisms and from, say, the Pacific to the Atlantic ones. These figures here come from this area of the Atlantic. So the same organism in two parts of the world's oceans is behaving very differently in terms of phosphate uptake based on the phosphate chemistry of the seawater that it's living in. And the smaller organisms are taking up much more. Why this is important, I'll move on to the next slide, is because as the world's oceans warm, we think there'll be more of this prochlorococcus around. And that's a very small one. And because it is able to take up phosphorus even at low levels and grow very successfully, it means that there will still be a lot of carbon dioxide sequestration to form carbon in the biomass of the microorganism. Typically, we think that, or we've been trained to think, that only certain parts of the world's oceans will be very productive, and other parts of the world's oceans will be rather low in production as the oceans warm. This is due to stratification and not enough nutrients reaching the surface. But if we get a shift in the population to the ones that can survive with low levels of phosphorus, then this importance of stratification is less important. And then we have the means of preventing a kind of a runaway situation of buildup of CO2 in the atmosphere. They'll still be able to photosynthesize and still draw down CO2 even under low phosphorus conditions. I went through that very quickly. I'm sorry. Happy to answer any questions on that towards the end. So we've gained a lot of knowledge. What are we doing with it? What is the responsibility? Well, one of the things is the algal industry here in Maine. This is a word cloud showing that essentially there are lots of opportunities around algae and algae development. So a algal cluster initiative has been created both around the microalgae and the macroalgae to sort of bring together the researchers, ourselves, Gulf of Maine Research Institute, University of Maine, together with the funders of seed corn funding, of stimulation funding, like the Maine Technology Institute, Coastal Enterprises Institute. And the two of these working together with the producers of both microalgae and macroalgae to start to open up new markets around algae, bioenergy, aquaculture, feed, chemicals, those sorts of things. So this Maine algal cluster initiative is just getting off the ground now. There'll be much more information coming out in the public over the next few months. And you can see that it's got some pretty ambitious developmental goals, but it is a unique partnership between the public sector funding, the research institutions, and the producers to try and really put Maine on the map with regard to algal initiatives. One of the other challenges that we have is on the high seas of the world's oceans. There's a bunch of acronyms up here. This stands for Genetic Marine Resources in Areas Beyond National Jurisdiction. I deliberately put it up as an acronym rather than spell that out because this is the world of the UN, United Nations, and the law of the sea. This is the International Union for the Conservation of Nature, and they have been working on this marine genetic resources issue for a number of years, trying to understand who these microorganisms belong to if they get transformed into biotechnological applications. In other words, drug discovery. And there's a little chart up here showing that you would need to collect a lot of samples to get just one or two examples of a breakthrough of a drug or a compound. Usually starting off with public funding, and that's why the UN is interested in this. Maybe it's the pharmaceuticals down at the end here, pharmaceutical companies who are investing in the final product. But this is happening. USA, Germany, patenting quite a few marine microorganisms now, followed by other parts of Europe. So this is happening on the high seas of the world's oceans, but we don't understand who this belongs to, where the proceeds should go. And that's part of what the UN has been working on. Just in case you thought I was making that up, you can actually go on the web, and this is obviously a very good product. It gets 4.3 stars out of 5. Alive marine phytoplankton. So 1 fluid ounce of this, I didn't put up the price, I think it was about $40 for 1 fluid ounce of a living concentrate of marine phytoplankton. You put a few drops in your tea in the morning. Actually not tea, that would kill it. Just in the water, and drink it. And it says here that it boosts your immune system and detoxifies you all in one go. So this, the marine products are definitely entering into our food chain for either pharmaceutical products, for something called nutraceuticals, which is essentially health skin care products and so on, shampoos, skin creams and so on, as well as industrial products like animal foods. So it's a challenge to understand the regulatory framework for that and to protect the environment at the same time. So I know I've overshot on the time, so the last 3 minutes are to tell you, or to invite you up to the public library here in Portland. We have an exhibit of images going on till the end of this month, till the end of March. There's an app. You can write that down right now and you can get a preview because you can look at this right now on your smartphone. And to close, I'm just going to show you some of the images that you would see if you went to go around this exhibit. So I think this will run just for a few more seconds, but I would like to thank you very much for listening to me. I'd be very happy to answer any questions.